journey from the lab to the clinic. We are going to be recording the meeting or the webinar, and hopefully we'll be able to host it on our website after this. You'll see that the chat function is disabled, but if you want to ask questions, you can click on the Q&A button at the top of Teams and enter your question in there, and those get sent to me. They aren't made public, so don't worry about asking, whatever you want to ask. And then I hope that we'll have time to address at least some of the questions, probably not all, to be honest, but at least some of the questions with Ralph at the end of his talk. So, without further ado, I will hand over to Dr. Ralph Hector. Ralph is a research fellow based here at the University of Edinburgh, working within the Symes Initiative for the Developing Brain, which is the sister centre of the Patrick Wiles Centre. Ralph has been very much involved for quite a long time now, dare I say, Ralph, quite a long time, yes, in research around genetic therapies, particularly for conditions like CDKL5 syndrome and Rett syndrome, the latter of which is one of those conditions in which this research is the sort of furthest forward, I would say, in this field of neurodevelopmental conditions. So, delighted to have you, Ralph. I will turn off my camera and hand over to you so you can share your screen and take us away.
Thank you. So yeah, thanks Andy and Sarah for asking me to give this talk. So I have just realized that I'm so used to Zoom that hopefully everyone can see this screen now. Can everyone see this screen?
Yes, we can see it.
Okay, if there's any technical issues, please feel free to interrupt and let me know that I am not presenting. Thanks for having me. This is a very unimaginatively titled talk. I'm sorry. I looked, when I was putting my title slide in, I looked to see what we'd sort of called the talk, and I simply just called it gene therapy. So as I said, not very imaginative, but it is covering, I guess, like sort of the basics of gene therapy and some of the work that sort of I've been doing in gene therapy over the last little while, what's happening in the field, some of the success stories, some of the drawbacks and so on with gene therapy. So hopefully this is going to be a sort of informative talk, whether you know a little bit about gene therapy or whether you know absolutely nothing about gene therapy. So yeah, as Andy said, there's a chat function. So if I'm going to go through all these slides and hopefully give a sort of good description about what happens in gene therapy. If there's any questions that you have during the talk, feel free to put in the Q&A to Andy and he can sort of interrupt me and ask me to expand on something. Or if you don't want to ask, I'll put my email address up at the end, and yeah, feel free to get in touch after the talk if you've got any questions. So, as Andy said, I'm based at the Centre for Discovery Brain Sciences at the University of Edinburgh. Full disclosure, I live in Glasgow and I'm Glaswegian. If you can tell from my accent. So I've been working in gene therapy for over 10 years now. I had a background in sort of genetics and then virology, and I was doing some sort of non-clinical related sort of postdoctoral research. And then about sort of 11, 12 years ago, I kind of realized that I really wanted to do something in the more with genetics and also in the therapeutic landscape. So gene therapy for me was a perfect fit, and it's something that I kind of like really sort of changed my career to work towards. So like I joined Stuart Cobb's group initially at the University of Glasgow, and then about sort of 7 or 8 years ago, we moved through to the University of Edinburgh to join the team there and set up a gene therapy lab. And we've worked mostly on Rett syndrome, which is a central nervous system disorder and a neurodevelopmental disorder. And we've also worked on some other syndromes as well. And sort of we've been sort of driving a lot of that work from sort of early preclinical research, so involving sort of cellular models and animal models, and then getting that towards the clinic. And in the last few years, we've been really lucky to partner up with an American gene therapy company called Neurogene, and we've been able to sort of get our gene therapy for Rett syndrome into the clinic. So I'm going to start with just giving a little bit of sort of genetics background, and then like what is gene therapy, and then hopefully kind of work towards some real world examples of some of the what's happening in the gene therapy world. So this is a sort of genetics 101 opener. I'm pretty sure everybody will have heard of these different phrases on the screen. So DNA, maybe RNA, and definitely protein. And this is just a little reminder, and it just kind of sets the scene for everything that I'm going to be sort of talking about in the upcoming slides. And the DNA, this is our genetic code. It has all these little functional units called genes, which are like the absolute cornerstone for making our cells work properly, for making our bodies work properly. And this DNA, this genetic code, is really the blueprint to then create the functional proteins that actually do the work in the cells. And you have this little intermediate product as well called RNA. So DNA is the sort of as I said, the blueprint. It is then transcribed into RNA, which is this little message, and then that is translated into protein, which is really the functional unit, functional element in the cell that actually does the work. So sort of the relationship between DNA protein is going to come up a few times in this talk. So that's just a little slide to kind of set the scene. And so the genome, that's basically all our DNA. In every cell, there's a full genome, which is basically all the DNA that codes for every sort of functional protein in the cell. And if you were to go down to the Wellcome Centre in London, which is just quite close to Euston Station, they've got this bookcase, and in this bookcase they've got a whole load of books, and within each book they've got the genetic code for a human. So this is a sort of this was made after the Human Genome Project, which was carried out roughly about 20 years ago now, in which, as DNA sequencing technology progressed, they were able to. The goal was to sequence an entire human genome, which is billions of letters of DNA, and each one of these kind of numbers represents the chromosomes. So you can see we're going all the way up here, and we've got the X and Y chromosomes here. So each one of these books, if you were to go to the bookshelf and take it off and look, it's just pages and pages of genetic code, lots of A's, C's, G's, and T's. And it's just to kind of show, it's just a representation of the genetic code and the genome that's in all our cells. And as you can might imagine, with so many different letters in our genome, there's going to be variation from person to person. And this variation can result in different things happening. Sometimes the variation has no effect at all, causes no difference. Sometimes it can result in some of the variation that we would sort of generally see by looking at one person, looking at another person. So for example, eye color, there's going to be changes in your DNA that are going to contribute to different eye colors. Then there's other kind of changes that are going to be sort of impact a response to things like diet and medication. So everybody would kind of respond slightly differently to drugs. That's why no drug works 100% effectively across every single person. People react differently to different drugs and to different diets, and that's partly genetically based. Then there's other variations that will influence the likelihood of disease. So there'll be certain changes that will sort of predispose some people more to certain diseases than other type of diseases. And then there's even more consequential changes to the DNA that will then directly result in a genetic condition. And in this example, they've got fetal ketonuria, which is always tested for at birth. And obviously, as everyone in this talk will know, that some of these changes can also result in some genetic disorders like Fragile X and SYNGAP deficiency. And somebody once described this bookcase to me as like having a genetic disorder is like opening up all these books and finding one typo in this entire bookcase of letters. And that's akin to sometimes what happens with some of these genetic disorders. It's akin to finding one typo in one of these books in this bookcase. So as I said, so these variations in the DNA can result in disease. So in a sort of normal cellular state, you're going to have your DNA, and it's kind of skipping the RNA step here, but it results in a functional protein which goes about its business in the cell, kind of works and performs its functions normally. Sometimes variations in the DNA, which are often called mutations, this can result in a non-functional or a missing protein or less protein or a truncated protein. And there's lots and lots of this is a non-exhaustive list of some of these DNA changes that you might see. So these are phrases that you might have heard of: missense, nonsense, frame shift, insertion, deletion, repeat expansion. These are all changes to the DNA that can then result in the genetic code changing. The RNA is different and the protein's different. And quite often, and again, there's exceptions to this sort of rule, but generally speaking, it tends to result in a sort of non-functional and missing protein, and that can contribute to disease and to genetic disorders. And some of these changes can be very sort of penetrant, and when I say that, that means that like one change in one gene would cause a disease, and we tend to call them single gene disorders. So one DNA change in one gene would directly cause that disorder. And so some examples here would be like spinal muscular atrophy, Rett syndrome, which I've worked on for a long time, Fragile X, SYNGAP. So these are, single gene disorders. There's also multigenic disorders, or sometimes called multifactorial. And this is normally where you've got multiple genes being involved, and quite often with environmental interactions as well. So this is kind of more sort of what you would maybe technically call low penetrant mutations, where you have this mix of predisposing and some protective gene variants, and each of them contributes like a little to predisposition to a particular disease or disorder. Example of is autism. There's lots of other conditions which are not caused by a single gene, but caused by a small effect of multiple genes. And these can be very complex, not only to understand, but also to then treat, just because you have these so many other factors affecting sort of how this presents in a patient. And in terms of, this is a gene therapy talk, and in terms of gene therapy, gene therapy is much more amenable to these single gene disorders, where if we know there's a single gene that causes this disease, then that can be our target. And fundamentally, gene therapy, you're trying to give back a functional copy to kind of replace or augment this broken or dysfunctional gene in the cell. Now, there are multiple sort of potential therapeutic strategies if you have a single gene disorder. So, there are conventional therapeutics, and by this, I kind of just mean every drug you can think of, like every over-the-counter drug, every prescribed drug. Ultimately, some can be effective, others cannot. Quite often you find that for a lot of these genetic conditions, there's a real wide variety of symptoms that each patient has, and it can vary significantly from patient to patient. And quite often these drugs are kind of used to sort of treat specific symptoms rather than the root cause. And so they can quite often have sort of limited effect. So I know that, for example, our experience with Rett syndrome, most of the girls who are affected, from girl to girl, they have a huge difference in the severity of the symptoms. For one girl, it may be that her breathing is the worst aspect, and she might be put on drugs to try and help the breathing. For another girl, it might be that epilepsy and seizures are the worst aspect, and she may be put on epileptic drugs. So it's all drugs to deal with very specific symptoms. And as I said, they can have sort of mixed effects, but fundamentally they're not curative. They're there to sort of alleviate some of the symptoms associated with each condition. Protein therapy is an option. Again, if sort of, again, just using Rett syndrome as an example, those the girls with Rett syndrome are lacking MECP2, which is the name of this gene and the name of this protein. So just giving back the protein could potentially be an option. That can be, proteins can be difficult to make, they can be difficult to deliver, they can be particularly difficult to deliver to the brain. These are all sort of disorders where we would be sort of like targeting the brain, or at least the sort of central nervous system. And also getting the right dose with protein therapy can be very, very tricky as well. So there are some therapies out there that have, like, for example, enzyme replacement therapy. Some work, but for some of our CNS disorders, protein therapy is like, has potential, but the technology is maybe not there to make it like a really sort of effective treatment. There's also stem cell therapy. Again, the drawback to stem cell therapy for brain conditions is that it's quite hard to get these stem cells back into the brain to be able to do their job there. I'll cover this again in a couple of slides. And one of the reasons that gene therapy is attractive for these single gene disorders is because we can maybe give back a functional copy of this single gene to the cells that are lacking this gene to try and treat this. So it's kind of tackling the root cause of the disorder that some of these other therapies, particularly some of the conventional therapeutics, are not. You're actually trying to replace the gene that hopefully then has this knock-on effect in the cell and tries to restore some of the homeostasis to the cell and get things kind of working again. It also has the advantage it's potentially a one-time treatment. So both for drugs that you would take for symptoms and also for protein therapy, you have to take that many, many times for it to work. You probably be taking it for the rest of your life. Gene therapy has the potential to be a one-time treatment and get the gene back working in the cells. And so obviously today the focus of the talk is going to be gene therapy. Certainly all these reasons are the reasons that sort of I got into gene therapy in the first place, to try and sort of get having spoken to so many families with some of these conditions, like gene therapy is one of the reasons why I wanted to use some of my genetics to try and get into this space and try and kind of help develop some of these drugs. So I was like using this image of gene therapy in talks for a couple of reasons. So one is that it kind of, I think, shows quite nicely that you're delivering DNA. The drug that you're delivering is DNA. And ultimately, if you think back to that very first slide, you're trying to get the DNA into the cell to make the protein to do its job in the cell, and that could potentially get things be really sort of therapeutic for the cell. The other thing is it's quite gold, which kind of gives the impression that it's quite expensive. In gene therapy, I'm sure all of you will have read a story about how expensive gene therapy is, and certainly in the moment that is the case, it's going to be an expensive drug. The one thing that this doesn't convey, or conveys wrongly, is that it's in the form of a pill, and certainly at the moment this cannot be given as a pill. We'll cover kind of how to get the DNA into the cells and into the body in a slide or two. So that's the one sort of misleading thing about this slide, but it does convey sort of getting DNA back into the body. And so I'll be talking mostly about gene therapy, gene augmentation therapy, in the sort of top left. I mean, that's the form of gene therapy that I've worked on the most, and really is the most advanced for a few different reasons. But there are other kinds of gene therapy out there, and you may have heard of some of these. So I just wanted to sort of touch base on these just before we move on to other parts of the talk. So in gene therapy, it can be called different things: gene augmentation, gene replacement therapy. Ultimately, what you're doing there is you're delivering the DNA that will code for the protein for this single gene disorder. So again, for Rett syndrome, our gene therapy is going to be encoding a functional copy of MECP2. We're going to get that into cells, and then hopefully we're going to get that's going to make a difference to how the cell functions. There are other types of gene therapy out there that have been in the news, have made really significant progress. Probably the one that most of you will have heard of is like gene editing, or CRISPR, as it's called as well, which is an acronym for one of the technologies. And this is gene editing. So this is actually, so with gene therapy, you're going in and you're adding this sort of functional copy of the gene to the cell. With editing, you're actually going in and you're either fixing the DNA or the RNA that has the genetic variation or mutation, and you're trying to fix that and then cause that bit of DNA or RNA to then make the correct protein. So you're just trying to fix it at that level, above the protein, and change the genetic code there so that it makes functional protein again. And that is exciting. It's made huge progress in the last sort of 15, 20 years. It's used a lot in the lab for genetic tools, and it's very useful and very effective. It's still not quite at the level where it's efficient enough to be used as a therapy across the board, but it's getting there and it's making a lot of progress. But it's still a little bit behind gene therapy in terms of maybe the ability to be used as a therapy. Like it's not been in as many clinical trials. The other sort of drawback that you have with with editing or CRISPR is that what you're editing is very specific to the genetic variation. So again, to use Rett syndrome as an example, there are lots of different sort of genetic variants and mutations that cause Rett syndrome. And the most common of these is what's known as the T158M mutation, and that accounts for roughly about 10% of all patients with Rett syndrome. So if you develop a CRISPR editing drug to treat that patient population, then that drug is tailored to fix that mutation at that specific point. And if you did have a successful drug, that could be great, but you're fixing 10% of the Rett population. And if you want to fix one of the other variants or the other mutations, you really are kind of having to make a new drug. So you really have to have like a specific drug for each of the genetic variations within that gene that would be causing the disorder. Gene therapy doesn't really care as much about the type of mutation or what mutations caused. It's really sort of applicable to pretty much any, I mean, again, there may be exceptions, but it's pretty much applicable to any child with, for example, Rett syndrome. So that's the advantage, certainly at the moment, that gene therapy has over like DNA or RNA editing with CRISPR. So I guess that's the distinction there. There are other kinds of genetic conditions where sometimes it's like too much of a gene is the disease-causing part. So one example I've got here is Huntington's, where there's too much Huntington protein being made rather than too little. And so there's other like forms of genetic therapy there. Some of these you may have heard of. That again, there's lots and lots of acronyms in the sort of genetic therapy world, but this is RNAi interference, or ASOs, which are antisense synthetic oligos. And these are used to sort of target this overexpressing or toxic protein, normally the RNA level, and then reduce levels to try and get back to a sort of non-disease state. So that's another form of genetic therapy. Most of the, certainly for Fragile X and SYNGAP, it's a loss of function rather than this sort of gain of function. But there are conditions out there where too much of a particular gene is the cause rather than too little of it. And then there's also cellular therapy, which I mentioned before. Sometimes this is also called ex vivo gene therapy. And this is where cells are removed from the patient. They're genetically modified outside the body in a lab, and then they're reintroduced into the patient. And, there are really significant success stories here. The biggest one is the CAR T therapy for leukemia. that has been a real success story. It is a particular case though where that this therapy is amenable to then a cell transplant going back in. As I kind of mentioned before, that's not as applicable um, for conditions where we're trying to, primarily target the brain. So these are kind of different forms of gene therapy that all have a place. But at the moment, certainly gene therapy or gene augmentation or replacement therapy is the kind of the most advanced. It's the sort of like the most likelihood of success at the moment. Some of these other technologies may sort of like catch up. Like there's again, as I said, there's been so much progress with editing and CRISPR over the last few years. There's new forms of that coming out all the time. So, this is good because it may be that in the future that we have multiple options available to be able to, treat these different disorders. But it's gene therapy that we'll be kind of focusing on today, and it's that's the sort of technology that I'd be kind of focused on with Rett syndrome over the years. So, what's in a gene therapy? So, as I mentioned, we're delivering DNA, and there are really only sort of three critical elements in a gene therapy that have to be there for your gene therapy to function. There's lots and lots of other genetic elements and lots of little sort of regulatory things that you can do to make your drug more effective. But at the absolute simplest level, you have to have three things. You have to have a promoter. You have to have your transgene, which is the gene that you're trying to express. And you have to have this functional element at the end of your DNA. It's I've put the technical name in here. It's the 3' UTR, which is like untranslated region, and it contains a poly, which is this polyadenylation signal. These three elements are at the basic level the critical things you need. So these are the DNA elements that you need. So the promoter kind of does what it says in the tin. It kind of promotes expression of your gene. It really helps to control where, when, and at what expression level the gene is switched on. So once it's in the cell, the promoter kind of will direct when the transgene is switched on and how much is made. And then you've got your transgene. So this is the DNA that codes for the therapeutic protein that you want in your gene therapy. So again, for Rett syndrome, it would be MECP2. For SYNGAP, it would be SYNGAP1. For Fragile X, it would be FMRP or FMR1 in the gene. So you're putting in the DNA sequence there of the gene that you want to make in the cell that's going to replace or augment the sort of the broken gene. And then this last element is really critical. So again, if you think about the DNA, the RNA, and then the protein, this DNA at the end is like really, really critical to make a good quality RNA that is then made into protein. So you have to have this extra element on at the end that kind of stabilizes the RNA, keeps it in the cell, and tells the cell to make it into a protein. And this can be modified as well. This can be modified in a few different ways to sort of like tune the level of the protein in different cells. And as I said, there's lots of other modifications that you can do to the gene therapy, lots of DNA elements that have specific purposes, trying to make it more specific to the correct cell type, trying to get the right level of expression. But fundamentally, these are the three things that you absolutely have to have in your gene therapy. And then, so say we've got a gene therapy. In fact, actually, one thing that I kind of just, I guess I wanted to add is that I'll show you a timeline in a few slides time where we kind of go through some of the like sort of the milestones as we got the Rett syndrome work sort of towards the clinic. Even in my time as a researcher, there are certain technologies that kind of come along quite quickly. When I first started doing gene therapy projects, making these kind of constructs was quite long and laborious. There was lots of, I'm sure you've all heard the phrase cloning. I had to do a lot of DNA cloning in the lab to make these, to then be able to start experiments. The technology has really come on. So, like, if I now was to design a gene therapy, I can really just order the DNA that I want from a company, wait a few weeks, that DNA part gets delivered. It really massively takes out the sort of logistical and technical difficulties of performing cloning in the lab. Cloning is still really commonplace. We still need to do it all the time, but actually, that's a bit of technology that has really kind of speeded up progress over the last kind of few years. So, say we've got our DNA, how do we get this into a patient? So we need a vector. So, and the vector is basically just a vehicle that carries this genetic material into another cell. So whether this is in the lab, whether this is in one of our preclinical models, or whether this is a patient, we need some sort of vector to get our DNA into these cells. Now, there are non-viral methods, so it could be naked DNA, it could be plasmid DNA, or there's other sort of like technologies like lipid nanoparticles to get these into cells. Now, some of these either are not particularly efficient, like naked DNA, or they might be efficient, but there still needs quite a lot of work to be able to get this to the point where it would be efficient enough to give it to a patient. So the real kind of efficient methods is viral. It's to use everybody's had, I'm sure, the cold or the flu. You kind of know how effective a virus can be at transmitting and getting into your cells. So we take advantage of these aspects of a virus, and we kind of manipulate it, and we kind of take out some of the sort of the disease-causing part DNA elements in a sort of standard virus and replace them with the genetic cargo that we wanted to deliver. So in this case, we would basically take out some of these sort of disease-causing parts of the virus, keep in some of the essential DNA, and we would put in the promoter, the transgene, and the poly sequences that we want to be then expressed in the cell. That then becomes a package. It's our DNA construct, gene therapy construct in a virus, and then we can deliver that to our models or to patients. Now, the advantage of these are that it's way more effective than some of these other methods in delivering genes into cells, particularly to the brain, and we'll cover that in the next couple of slides as well. There's lots and lots of experience. Actually, this is an old slide. There's now been thousands of patients dosed with viral vectors in lots and lots of clinical trials. And although it's still a relatively young field, we know that it can be long-lasting. So a single dose of a virus can result in long-lasting expression of our gene therapy. There are obviously, like all technology, some disadvantages. There's only so much capacity, there's only so much space we have to get the DNA that we want into these viruses. So for really large genes, that can be quite a challenge, a technical challenge. But for other disorders, that doesn't matter. But it is one of the considerations, particularly when we start working on new disorders, like can we actually fit the replacement gene into the virus? They are complicated to manufacture. As you can imagine, trying to make large, huge quantities of virus that are high quality enough to be injected into patients is quite a consideration. Withdrawing the treatment isn't possible. This is as we talked before about conventional therapeutics or even protein therapy. These kind of wash out the system. They need to be then re-administered, whether that's every few weeks, every few months. Once gene therapy's in, it's in. You can't then withdraw it. The virus has got in, it's got into the cells, it will be expressing the transgene. So that's quite that's an advantage as well as a disadvantage. And it's why kind of safety experiments are so essential. But yeah, you can't withdraw the treatment. And there's also potential for immune response. As you can imagine, with delivering large quantities of virus to a patient, there's the potential for significant immune response there. So the virus that we use primarily for gene therapy and particularly for brain disorders is AAV, which is this adeno-associated virus. So just if you see AAV, it's really at the moment the sort of gold standard of viruses that would be used to deliver gene therapy to most cells in the body. I think particularly for the brain, there's different forms of AAV. Technically, they're called serotypes. So there's different naturally occurring serotypes of AAV that are really good at getting into different cells in the body. AAV has been used now for several years to try and be an effective gene therapy, and probably the best known is you may not have heard of this, the drug name, but it's Zolgensma. So this is an approved AAV9 drug that's used to treat spinal muscular atrophy, and I'll talk about spinal muscular atrophy in a couple of slides' time. But well over 3,000 patients have now been dosed with Zolgensma. So this is a delivery method that can get into the cells in the central nervous system. The main target for spinal muscular atrophy is the spinal cord cells, and AAV9 is really effective at getting into these spinal cord cells. Because there have been so many patients dosed, it's got a known safety profile. Now, that doesn't mean it's 100% safe. It just means that there is a known safety profile. So there are certain doses that cause immune reactions. There's a little bit more known than, for example, other viruses which have been used in the past to sort of deliver gene therapies. And it's still the virus of choice for lots of clinical studies that are underway and planned clinical studies in the central nervous system space. So it's still the gold standard. And actually, AAV9 is the vector that we use in our Rett syndrome gene therapy trial that's currently ongoing, just simply because currently there's not a better vector out there that enables us to get our gene therapy into the right cells in the brain. So we've got our DNA construct, our gene therapy construct, and we've packaged it in a virus. Now, how do we get it into the body? Now, as I said, we're particularly focused on the brain, where we really want to get this treatment to. And there's multiple ways to get it there. There's some quite invasive methods to get it there by direct brain injection. So there's the ICV, the intracerebroventricular route, which kind of targets the ventricle in the brain. And then there's the ICM, which is the intracisterna magna, which is this kind of like space at the back of your sort of the bottom of your skull, just behind there. There's also intrathecal lumbar route, which is kind of in the base of your spine. And the reason that these are sort of preferred methods is because they bypass the blood-brain barrier, particularly these methods, ICV and ICM. AAV9 kind of likes to get into brain cells, but it's not very good at passing through the blood-brain barrier. So if you were to give an intravenous injection, which is obviously a lot less invasive, then it would go around the body, it would go through the bloodstream, a lot of it would go to the liver, not very much of it would get past the blood-brain barrier and into the brain. And if your main target is the brain, then IV isn't necessarily the best route. And so again, to use our Rett syndrome gene therapy trial as an example, we chose the ICB, the direct brain delivery route. And that was partly based on, I've really tried not to show any kind of data in this talk, just so that it's just not, it doesn't become like a big long data science presentation, but a study that our sort of gene therapy sponsors, Neurogene, performed was basically looking at these different delivery methods and how much gets into these different brain regions by using these different routes of administration. And I guess just by ignoring these numbers, if you just look at the higher the graph goes, basically it's like more of the DNA is getting into these brain regions. And I guess what you can see the trend is that if we deliver by ICB or ICM, which are these like direct brain delivery routes, then you get more of your gene therapy into these different regions of the brain, and particularly when you're trying to hit sort of like either get into several brain areas or you're trying to get into some of these sort of deeper structures like the cerebellum. It's kind of really important to be able to kind of maximize the chance of your gene therapy working. So that's why there are kind of such invasive routes. For spinal muscular atrophy, for example, there were some studies with intrathecal lumbar and actually intravenous. You can get each sort of hitting off cells in the spine with either of these routes. But for Rett syndrome, for example, we know from a whole bunch of like studies that were performed by us and other groups that to maximize the chances of it working, you really have to hit some of these brain structures like the cortex and the cerebellum. And for that, you kind of need to go into these more direct routes. So, having said that, the reason we're doing that direct route is because AAV9 is the current gold standard out there for trying to get our gene therapies into the brain. And the but there's pretty major efforts underway to find better vectors. There are entire companies now devoted to basically developing vectors that are better than AAV9 at getting your gene therapies into the right cell. Um, this is sort of a combination of things. This is like an exploration of like naturally occurring viruses to see if they will work better. And then there's a whole bunch of sort of design, so like rationally engineered, directed evolution, AI design. Like these are all sort of different methods that are being employed by labs and companies to try and basically just get better, more efficient vectors. If you have vectors that are more efficient, you can bring the dose down, which increases safety. So these are all really important things that would really benefit the gene therapy field. And there's a real concerted effort to try and get better vectors. And I think this is a space that's going to really change in the next sort of like 5 years, for sure. Every time I go to one of the gene therapy conferences in America, there's been more and more progress with more efficient vectors that you can, ideally deliver IV. So as I said, like if you can deliver IV, it's much less invasive. You don't need surgery. There's so many aspects of that would be so much better if you could deliver that IV. So if we can get these next generation vectors to be more efficient when delivered IV, then that's going to make a real difference to be able to, even for clinical trials, like not having to put patients through some of these invasive delivery procedures, and just make it safer as well. So like more targeted to the tissues and cells that we actually want to target, and less targeted to sensitive tissues and cells. And that's going to be that's like a major goal of some of the companies who are kind of developing these vectors, and also trying to lower the potential for immune response as well. And that's kind of one other thing that I just want to kind of mention here. So, undoubtedly, immune issues are a challenge in all gene therapy programs, and probably there's no exception. But immunosuppression treatment, it's probably not just common. It's probably probably happens in I would say in probably almost all gene therapy treatments. And it's a very sort of complex field, and the field is trying to get more answers to this. It could be a response to the virus capsid going into the cell. It could be a response to the virus DNA. It could even be a response to the protein that we're making in the cell. There's lots of work being done on this to try and make this safer. I mean, this can also work against gene therapy, because if there's any sort of pre-existing immunity to viruses like AAV, then that can really impact the effectiveness. So for the most part, patients are screened. So if patients going on a trial, if they had high pre-existing immunity to AAV, it really might render that gene therapy ineffective. It would just block it from getting into the right cells and like really impact how effective that would be. And immune responses can also kind of limit the ability to redose gene therapy. It looks like there's a lot of data that kind of supports the view that there would be like a single dose would be kind of long-term expression. But as I said, the field is still relatively young. A lot of these like trials have started in the last few years. There's going to be a lot of data coming out over the next few years, 10 years of patients who've been treated with gene therapy, and we're going to get a sort of much better insight into how long these gene therapies express for. It may be that they may need topped up at times, so that's why again there's efforts underway to sort of allow kind of redosing. And so on that same sort of note, is it really kind of a one and done? There's so many other drugs and therapies that have to be taken regularly to be effective. But is gene therapy really a one and done? Well, most drugs are eventually kind of washed out of the body, whereas gene therapy kind of delivers a sort of permanent drug to the cell. So in this little diagram on the right, it's kind of showing a cell, it's showing your endogenous, your own chromosome in there. And what the gene therapy does is it delivers this little bit of DNA called an episome. So again, in the case of Rett syndrome, what we're delivering there is almost like what we call a mini MECP2 chromosome. So it's not integrated into your DNA, but it sits in the cell and it expresses MECP2. So it really kind of becomes part of the cell without integrating. And again, in terms of the brain, mature brain cells don't divide, so you don't get this dilution effect that you maybe get in other cell types. And so that's why, certainly for some of these, central nervous system disorders, there's a pretty good chance that we would have sort of a long-term beneficial effect from a single treatment of from gene therapy. And as I said in the previous slide, it's still relatively young field, and there's not too much in the way of like really long-term patient data, but that's gonna come in the next few years, particularly as I said, when you have success stories like the spinal muscular atrophy treatment, where you have several thousand patients dosed, that's going to be really, really important information for the gene therapy field over the next few years. And the spinal muscular atrophy treatment is probably the poster child for AAV and for AAV9 gene therapy. It's a really severe disorder. The kids have motor significant motor deficits and breathing difficulties, and a lot of the children die between 2 and 4 years old. It's a really, really severe genetic disorder, and it's caused by loss of function of the SMN1 gene. And Zolgensma is the sort of the brand name for this AAV9 drug that delivers a functional copy of that gene into the spinal cord. And it really has been, a kind of amazing success story. There are thousands of children who are still alive and walking and breathing unaided, with a single dose of the Zolgensma, the gene therapy. It literally has saved lives. So, I urge you to go and read more about Zolgensma. It's because there's been so many patients dosed now, we're learning a lot more. It really does look like, for example, for this particular condition, that the earlier you can get a diagnosis and the earlier you can get a treatment, the more effective the treatment is. It, you know, it, There's sort of the idea that gene therapy might not always be completely curative, and like looking at the data for SMA, the closer you can get to the sort of early delivery of the gene therapy to these children, then the more profound effect it seems to have. So, and that kind of window of opportunity may differ from disease to disease. It may do. It may not be the case that for every single genetic disorder, the earlier you go is the best. But certainly for SMA, that seems to be the case. And it's a single dose. It's delivered by intravenous injection. and enough of the virus gets into the spinal cord to be able to make a really significant difference. So, this has been a real sort of groundbreaking gene therapy success story. And as I said, it's an AAV9 delivered drug. So I'm going to switch a little. I just talk a little bit more. I realise the time. So I'm just going to. I've only got a few slides left anyway. I just kind of wanted to give you a little bit of sort of our experience with Rett syndrome and getting this to the clinic. So I joined Stuart's lab about sort of 10 years ago, and I really wanted to work in genetic therapies. And Stuart's lab was really focused on understanding more about Rett syndrome. And his lab was funded by the Rett Syndrome Research Trust at the time. And this is Monica Conrads, who's the founder of RSRT. And this is her with her daughter Chelsea, who's Rett syndrome. And when her daughter was diagnosed with Rett syndrome, she didn't really know anything about it. She then sort of devoted her life to learning more about it and then setting up this trust that funds research um, into both understanding the disease and also like trying to find a cure, trying to get this into the clinic. And I've already mentioned this several times earlier in the talk, but this is a single gene disorder, the gene MECP2. If there's loss of function mutations, you get Rett syndrome. And there's a huge variety of symptoms that are caused by Rett syndrome, hugely variable from girl to girl. There's lots of sort of genetic sort of complications with Rett syndrome as well. And it's a very severe disorder, but it's potentially amenable to gene therapy because of the single gene disorder. And I kind of just wanted to show you a timeline. So rather than sort of just show a whole bunch of data, I just kind of wanted to show you a timeline. And I guess if any of you get any questions about this, or like please feel free to kind of follow up and email me if you've got any questions. But this is just kind of showing the timeline for getting the work done and getting this into the clinic. So in 1999, the link was made between MECP2 and Rett syndrome, and it was identified that MECP2 was the cause of Rett syndrome. And just a few years after that, there was a lab in Edinburgh, actually, Adrian Bird and Jackie Guy in Adrian's lab. They developed this Rett knockout mouse model. So this is basically where you take a mouse and you very, very specifically take out a copy of the MECP2 gene. So you have this kind of knockout effect, and they were able to recapitulate a lot of the symptoms of Rett syndrome in this mouse model. Now, having these models is really, really, I can't emphasize enough how important this is to be able to do the work to find out if your gene therapy is both effective and safe. So that early work was like really critical to say, well, here's a model, and by knocking that gene out, you're able to recapitulate some of these symptoms. And as I'll mention later, that sometimes a mouse is a mouse is not a human. Sometimes knocking out some of these genes has very little effect on symptoms and phenotypes, and it doesn't look like the disease, and that can curtail progress because you need to have a model to be able to kind of show that your gene therapy is actually working. You need to do this early work before making the leap into testing this in humans. And then what was really critical as well was a few years after this, they made another genetic model where they showed that Rett syndromes were reversible. So basically, without trying to go too technical, they made a conditional mouse model where they could switch on or switch off MECP2 expression in the mouse. And what they were able to show was that if the mouse started to show symptoms of Rett syndrome because it didn't have MECP2, by switching it on at a point in development, those symptoms were rescued. And that really kind of set the scene for gene therapy being a plausible therapy for Rett syndrome. There are Rett-like symptoms. and then switching the gene on, restoring the gene in those cells, helped to restore sort of like normal function of those cells and reverse those Rett symptoms. So that was like a really, really critical experiment that was done again by Jackie and Adrian in Edinburgh to kind of show that that's the case. And there's still a lot of conditions where this kind of reverse. It's not an easy experiment to do, but there's still a lot of conditions where that sort of reversibility or that sort of conditional mouse model is not used. But for Rett syndrome, that was absolute key to kind of show that restoring MECP2 expression in cells could really make a difference. And so shortly after that, there was the sort of the first proof of concept gene therapy studies. So when I say preclinical, I just mean anything that precedes the clinic. So this is like cellular models and mouse models of Rett syndrome. It was just shortly after this actually that I joined Stuart's lab and kind of was involved in a lot of the preclinical studies that were sort of bit by bit improving the efficacy of our gene therapy and safety. One really kind of important thing, which I don't want to go into too much detail, but the Rett syndrome has a sort of like Goldilocks like feature to it where not enough MECP2 causes Rett syndrome, but too much MECP2 is quite toxic. So we were constantly finding that with our gene therapy experiments that we were trying to get this sort of Goldilocks zone of MECP2 expression where we got efficacy and safety, but we didn't push into this sort of toxic zone, and that was very difficult. And we came up with like a sort of with a genetic regulatory system within our gene therapy product that made it much more efficacious, made it much safer. That took quite a bit of time, but we have the technology now. We can potentially apply this to other sort of dosage sensitive conditions. But there was like lots and lots of preclinical studies, and that was the technology that for us like came out as being the best technology to apply to Rett syndrome. And so once we kind of got much better efficacy and safety, we started putting together this package of data. Neurogene, who we partnered with, an American company, so we approached the FDA, and they also approached the MHRA in the UK to prove clinical trials, and then in 2023, clinical trials were initiated. So I just really wanted to show this timeline, just I guess just to show some of these milestones. Now, it's not going to be the same for some other genetic conditions. It's going to change. As I mentioned before, some of the technologies come a long way. Making mouse models is much quicker than it used to be. Making DNA, making virus, it's much quicker than it used to be. And just so many lessons learned mean that you hopefully are not making as many, unknown decisions with unknown outcomes sort of earlier in some of these studies. So, I think these timelines can be significantly shortened, but it was kind of just to kind of highlight like what it took to come along to get these into the clinical trial. So the company Neurogene that we partnered with in Edinburgh, we have a drug called NGN-401, which is our gene therapy product for Rett syndrome. And the classic, you wait in a bus and no buses come, and then two buses come at one time. There's another company as well called Taysha, who at the same time were developing a gene therapy, and pretty much at the same time came on with a clinical trial for their product, Taysha 102. So for Rett syndrome, there are now 2 clinical trials ongoing with gene therapy and constructs aimed at trying to help or ideally cure, but like really significantly help all these children with Rett syndromes. So this is just to kind of point you towards some reading. It took us a while to get our manuscript out, but we have this is recorded. Also, feel free to email me if you want any of this reading material. This is, our lab is involved in a lot of the preclinical work, so this is kind of detailing some of the work that we had to do in the lab. It really sort of encapsulates a lot of the work that had to be done so that we could convince the FDA that we had like sufficient, efficacy and safety that we could take that drug and take it from these mouse models of Rett syndrome and then take it towards the clinic. And then there's also information about the clinical trial, which is a phase 1/2 clinical trial that's that started, it's ongoing, it's recruiting in the US and the UK, and they're testing this for efficacy and safety. So, so again, like for us, it's another good news story. We've kind of, we wait to see the outcome of the Rett syndrome clinical trial. You can also go onto the Neurogene website and get updates there. I also don't quite often know any more than the press releases, because everything's kept quite confidential with the clinical team. So yeah, you can get some of these press releases from the Neurogene website about like the progress of the clinical trials. I mean, there are, I've mentioned this before, but all these kind of first in human trials, they're all safety trials. We're looking for efficacy as well. They're all safety trials. And gene therapy has significant potential. There are obviously inherent risks. As I mentioned before, it is an irreversible treatment once administered. There's often immune responses. It's common in gene therapy. That we try to mitigate against this by giving immunosuppressive drugs at the time of dosing, but it's still a risk that is common in gene therapy. Overexpression of your gene can be problematic. With MECP2, we knew this was problematic, and we had to work really hard to find a genetic system that enabled us to sort of get past that. So for Rett syndrome anyway, it looks like we've got kind of a system that can kind of overcome that, but it may be an issue in some other disorders as well. And there can be other unexpected effects, and also AAV immunity that can also contribute to sort of a less kind of potent efficacy effect from your gene therapy. On the flip side, risk of no treatment is also very high. Not doing anything if there's a gene therapy there, it's a significant consideration that as these therapies become available, it becomes a big decision for a family as to whether to enroll for a clinical trial or not. And if the trial, even if it's successful, there are still these inherent risks with gene therapy. So there's going to be a lot of decisions for kind of families to make in the next few years, whether it's a trial or even for an approved drug, as to whether they kind of take this on. And the other thing, again, this somewhat related to the sort of the redosing question, is this first generation of any therapy, it might not be definitive. Companies are always working in the background to try and make a sort of more effective drug. At the moment, redosing is very difficult. So if you've got the first generation of the drug, you may not be able to get the second generation of the drug, even if that's proven to be more effective. So that's the kind of a lot of the the things that need to be thought about if you're a family who is presented with the option of potentially enrolling in one of these gene therapy trials. I've just got a couple of quick slides left, I guess, just to sort of like touch more specifically on Fragile X and SYNGAP. The status of gene therapy in both these conditions is a little bit unclear. The idea of like sort of gene therapy for Fragile X, there's been some publications in the last couple of years from the David Hampson lab, and who was working with Taysha. I think Taysha kind of focused mainly on their Rett program now. But this actually, we had a PhD student in our lab who was also working in gene therapy for Fragile X, and her data sort of correlates with what David saw. So there is potential there. It does require a company to come in and just kind of get this, and sort of take these results and start building this package of experiments of efficacy and safety to sort of take this towards the clinic. And the status of some of these other companies who are involved in this, like RegenX Bio and Lysogene, it's a little bit unclear. Like our PhD student, they were aware that there are limitations of some of the preclinical models with Fragile X. There's potentially some issues with overexpression as well. The field's a little bit divided, and also some feedback we've got as well is that there is a complex clinical pathway, just in terms of the the variation in sort of symptoms and severity of symptoms, making a clinical trial to assess a gene therapy becomes quite a complex task. Um, but there's obviously still significant potential for gene therapy for Fragile X. And for SYNGAP as well, there's real potential, but again, it's sort of difficult to know where, um, what the state of play is there. Uh, there's Praxis Precision Medicines who I think are pursuing a similar approach to Stoke. Their approach isn't sort of a standard gene therapy augmentation approach, but rather they are using targeting the RNA, and they're using one of these antisense oligonucleotides. They're targeting the RNA in the cell to increase protein levels to get the cell back up to sort of like normal levels of SYNGAP, using like this ASO approach. Um, so the status of the SYNGAP work is not totally clear. They are currently in the clinic with an ASO drug to treat Dravet syndrome, so it's possible that that's where most of their efforts lie just now. The fact that they've got that and it looks to be effective for Dravet is a real bonus. Um, what's not clear is whether they need basically a new drug for to target SYNGAP, which I suspect they do, which is not a insignificant undertaking. But yeah, certainly a potential route forward. And again, like we had a PhD student in the lab who was working on SYNGAP gene therapy, and her project was complicated again by sort of some of the limitations of the preclinical models, and also some of the expression, some of our gene therapy products is sort of a complicated design for gene therapy molecules for SYNGAP, in the way that sort of like some of the stuff for MECP2 was a bit less complicated. But yeah, so that's sort of like a quick update. I realize I've probably gone over longer than I meant to. Hopefully that still leaves a little bit of time for questions. Feel free to email me. I hope this has been useful. If there's anything that I've not sort of expanded on, that you want a little bit more feedback from, or a little bit of pointers in the right direction for reading, then please email me, or email Sarah or Andy, and they can put you in touch with me.
That's great. Thank you very much, Ralph. That was excellent, and it was very informative and taking us right from your genetics 101 through some pretty complex concepts, right up to talking about sort of where things are at for some of these conditions. We have, if people have questions, there's been a few that have come in, but if people have questions, please do type them in the Q&A box, and I will go through and read some of them out. I mean, we also had some questions submitted before, as people registered, and lots of those really related to, when will this be available for specific conditions? And thank you for touching a bit on Fragile X and SYNGAP. I mean, you kind of answered that it's complicated. It's not a straightforward single pathway. I'm not going to ask you to commit to a number of years, in any way, state or form, but I mean, yeah. Is there a feeling in the field that there are going to be movements, not necessarily for, say, Fragile X or SYNGAP, but for other neurodevelopmental conditions? Rets is obviously the most advanced, and where things are in clinical trials. But I suppose, just speaking generally, there's the feeling that these are therapies which are more broadly generalizable, and that there will be
Yeah,
other.
I think there's going to be certain lessons learned and certain experimental designs and packages that will become a little bit more commonplace. At the moment, it doesn't feel like a little bit like every person for themselves, and everyone's getting their own package together and getting feedback from the FDA individually.
Mhm.
So I think as more and more companies go forward, I mean, we know that our experience is we've absolutely learned lessons for the next, package that, we'd be taking forward to the FDA for approval, for sure. And I think that, as kind of we publish and what we've done, there's just going to be a general sense in the field, like what should we be trying to do to make these packages more efficient and more effective and more plausible to take to the FDA for approval to get into the clinic. I mean, one thing that is, to be totally blunt, what is really difficult is once you're in the clinic, it tends to take up a lot of capacity. So we know that, for example, like Neurogene are in the clinic, it takes up a huge amount of the company to get that clinical trial going, and and other things kind of slow down. It's just the nature of trying to get these things done. So that might affect some smaller companies than bigger companies, for example, who've got other packages coming through. The other thing that I guess is like a little bit frustrating, probably for families, is not getting any insight into the progress that's being made. And the honest truth is that we were lucky that when Neurogene came along, they kind of partnered with us rather than taking in our technology and then doing it themselves in a black box that we had no access to. But even with that, like, we still then couldn't transmit all that information to families that we knew, because at the end of the day, these are companies. They're trying to get these forward, and sometimes there is a little bit of a black hole of information, and you don't know if there's significant progress being made or if there's no progress being made, and it's just one aspect of the sort of the industry that is sort of difficult to overcome at the moment. People don't want to be kind of updating all the time on like small incremental progress or no progress. So that becomes a little bit tricky. So, like, for example, when I was looking at the status with Fragile X and some of the SIMGAP programs, it's really difficult to know whether they're, a month away from submitting a package to the FDA, or if they've hit a problem and they're, a year back. It's just, those are the frustrating things in the sort of the timeline. But yeah, as you said, like, as technology and as these gene therapy packages become more commonplace, for sure, there's going to be so many more lessons learned about what should be in a good package to take forward to get approval for a clinical trial.
Cool. And a few people have asked about age of when people might have to have these therapies. What would be the best age for effectiveness?
So that's a really, really good question. Ultimately, this is not trying to pass the buck, but it's really going to be up to whatever companies take Fragile X or SYNGAP therapies forward to the clinic. It's going to be up to them to kind of set what they think is the most effective age that they will be able to dose, like, patients at. So I know that, for example, I sort of touched on this with spinal muscular atrophy, but those children they don't live that long. But what is clear from the clinical data is that the earlier they can get the genetic diagnosis and the earlier they can get the therapy, the more profound the outcome. So for spinal muscular atrophy, it is just as early as you can get the delivery in. For Rett syndrome, we think that there's a kind of window where, like, a pretty reasonable window where if you can dose kind of young children or adolescents, you still would be able to get like a really significant therapeutic effect. We think that the younger you go, the more effective it will be. In the clinical trial, we're allowed to dose patients between 4 and 10 years of age. And that's always a conversation with the FDA. So the companies can propose what they think might be the effective age range to aim for, and then the FDA will come back with another suggestion. They'll either agree or they'll shift the goalposts. Quite often, the FDA, and I assume the MHRA as well, will maybe for the very, very first clinical trial, maybe ask to dose older patients as a kind of for various reasons, but like for the Rett syndrome trial, like it's initially 4 to 10 years old, and that's because we think that'll be effective. We think it might be even more effective at a younger age, but they want to see efficacy and safety data at that. So I'm kind of going on a bit, but basically it's going to vary from condition to condition, and it's going to vary on like what the companies want to do and their interaction with the FDA and what ultimately gets approved. So it's not necessarily going to be the case that the younger is better for every single condition, but certainly for SMA and for for Rett, it seems like the younger you can dose them, the better.
Yeah. So from what you're saying, it's going to differ between different conditions, and at the moment we don't really know enough from most of these conditions, even from the lab studies and things like that, as to whether or not turning this gene back on in an adult laboratory model is as effective as turning it back on in a child. My understanding is there's not, just for a lot of the conditions we work with, that those studies haven't been done. Not sometimes because people haven't wanted to, but because they're complicated and the difficult to each own condition carries its own set of complications for doing them.
Yeah.
That's my sort
Uh,
of simplified understanding of it. Is that
yeah,
fair?
100%. Yeah, yeah. They're complicated experiments to do. We were kind of lucky that in Rett syndrome there was a lot of these models made where it was maybe more kind of finely assessed. And again, in mouse models, like translating that up, it's the best that you can do to make predictions. But yeah, but there was a lot of kind of fine precision experiments done and it's not the case for every disorder. So we were armed with a little bit more information there than other disorders.
And there's a couple of kind of practical questions, I guess, about dosing. One was you
Yep.
mentioned those kind of intraventricular, dosing directly into the brain. Presumably that's done under general anesthetic for people. And then the other was about giving immunosuppression. I think corticosteroids and things like that are giving immunosuppression. Is that just at the time of dosing, or is that something, like transplants where you have to give it longer term?
So generally, again, there's exceptions to this, and there have been some, really extreme immune reactions to the virus, and that it is just a known risk of gene therapy. Most of the time, no matter which route of delivery you give the gene therapy, it gets to the liver.
Mhm.
That's just, whether you're giving it directly to the brain or you're giving it in the vein, a lot of it goes to the liver. And there's other cells that might be affected as well, but the liver in particular gets affected by these doses of AAV, and you tend to get liver enzymes and immune responses there. So people don't tend, and they are generally, they overcome them. There's like a transient sort of immune response, and they're overcome. So they after gene therapy, you don't have to stay on these drugs for as long as you would, as you said, Andy, from like a transplant or something. It's more short term. It's to get through that initial thing where the patients have been subjected to essentially a massive dose of virus, and it's waiting until that's cleared, things have settled down, and once things settle down, you've got the virus is in the right cells and kind of working, and the sort of the immune response becomes less of an issue. So it's just that it's a short term thing, but it's very much a safety thing just now, just to make sure that there's not um, a significant response.
Okay, fine. Um, and then, I mean, this is a question that's very specific to Fragile X syndrome, and I've actually been asked it in other sort of forums before. But people worry about risks and worry about giving these medicines to their children. And I'm sure you're probably aware that in Fragile X there's a group of people who would be called premutation. The parents often of children who are affected by the condition, or adults affected. And there's a question that's asking, could it be tested on the people with this premutation rather than those with Fragile X syndrome? I mean, I suppose I will give my answer, which I've said before, which, but feel free to disagree. But the premutation is essentially, although it's a related genetic condition, the effects that it has are very different. You don't see the turning off of the gene in the premutation. So therapies designed for folks with the premutation are going to be very different from those designed for Fragile X syndrome. I don't know. I can see you nodding, but it's just if there's anything you would add to that.
Yeah, I mean, it's anytime you change anything in your drug, it's a new drug. So, it's literally double the work. So you wouldn't, you know, ideally be having even a slightly modified drug that you'd have one patient population
Mhm.
versus another. And I guess the other thing is you ultimately want, I mean, these first in human safety trials are designed to be safety initially. So I guess there's like a theory that you could maybe take the drug into that patient, that group, are they really a patient group? I mean, in a way, it's a very, very complex question to try and answer. I mean, sort of, it wouldn't be an efficacy study, put it that way, because if there's kind of nothing really to fix, then it's not really an efficacy study. It would be like a safety thing. But I, I would say that that would be, I'd find that unlikely to get past the FDA, put it that way.
Mhm.
I mean, like, and I guess just to, I don't know whether it's related or not, but I'll say it anyway. But like for example, for our, in classic clinical trials, you have placebo control group, which is a group of patients who, don't receive the drug or receive like what they think could be the drug, and they're not told whether it's the drug or whether it's like an inactive drug or nothing. That's not in, for example, the spinal muscular atrophy trial. It's not in our Rett syndrome trial. And that's unusual, but the reason that they don't do it is because, well, a couple of reasons. One is we've got a very invasive procedure, so they're not gonna pretend to sort of give a drug to the inside of the brain and then give a placebo. And then the second thing is it's rare and it's severe. They really don't want to have a group that is going to be told you didn't get the drug, particularly if it became if it was effective. So there's always caveats to these gene therapy trials. There's not a one size fits all for these trials. So I guess there's potential movement in there. But again, it's all down to sort of what makes the best clinical trial design, and then what your interactions are with the regulatory bodies to say, do you agree with us? And if you don't, what do you want us to modify that would make you happy? And then that's what you have to sort of agree on going forward into the trial.
And then the final question, which is a bit related to that, is how much are companies sharing information and things like that with each other to try and accelerate progress? Because there is a lot of learning that's going on, in this and other trial related fields. So, I mean,
Yeah,
companies, I guess, and academics and universities and things would be the other group of people who are probably sharing information.
yeah. So I mean the blunt answer is companies really don't share information. They're not very good at it. But I mean, there's reasons to not share it as well. I mean, the academic groups are better at kind of sharing information for sure. You go to a conference, and the American Gene Therapy and Cell Therapy conferences in a few weeks, and there's going to be a lot of information shared. There's something like 1,500 posters and 5 days of talks. There's going to be a lot of information shared. Companies don't tend to share, certainly between other companies, because ultimately they're kind of, vying for the same therapeutic space.
Mhm.
And they have businesses and employees and shareholders they kind of want to be. I mean, there's all these classic things of, like it's good to have healthy competition, but ultimately, like, companies don't want to be giving away some of their know-how. And that's just unfortunately a reality, because it'd be nice to think that there'd be a sort of halcyon world of companies kind of combining and getting therapy. I mean, if it one thing I would say that if this is sort of a little bit more of a sort of silver cloud on that answer, is that when the the Rett Syndrome Research Trust funded our work all those years ago when I kind of joined Stuart's lab, they did an unusual thing and formed consortiums. And those consortiums were, so our gene therapy consortium was three different labs working in Rett syndrome gene therapy. And they put us together, and we had 2 or 3 meetings a year where those academic labs came together, and we worked and we sorted problems and we shared reagents and we shared things that worked and didn't work. And a few years later, some of that work has gone into the Taysha gene therapy. Our work's gone into the Neurogene gene therapy. And there was even actually another company called Avexis who ended up not pursuing the Rett syndrome work, and but initially that was going to be a Rett syndrome. So even though at the company stage it can seem a bit bleaker and people don't talk, people don't collaborate, there is still really good work going out there and people do collaborate. And like I said, those academic collaborations those years ago, that resulted in 2 and 3 gene therapy programs coming along. So, the silver lining there is that, that early work, which is so critical to making the products that are going to ultimately get into these trials, there is good collaboration there, people do want to help each other, and it makes a difference to getting these things ultimately into the clinic.
Yeah. No, absolutely.
So
Okay.
I turned that from glass half empty into glass half full.
Well, no, when you were going glass half empty, I was going to exactly just point out that actually most of the collaboration, the sharing goes on at that fundamental science stage of things. And also actually, as you know, I work in clinical trials of non-genetic therapies at the moment, but as these results come through, these companies publish the results. There's often it's clinical academics who are working on these trials, and they are then learning from those, and that learning goes into the next generation of trials that get started for these conditions. And we've seen that very much in the Fragile X field,
Yeah.
moving forward. So there is a point whenever, for commercial reasons, information isn't shared, but all around that, actually, there is an awful lot of collaboration, and
Yeah.
that's the way science kind of works now,
Yep.
basically.
Absolutely.
Okay, that's perfectly taken us to the end of our time. Thank you
Cool.
again, very much, Ralph, for
No
an
problem.
excellent talk. Thanks to Sarah Ely for organizing all of this. Do feel free, people, to get in touch with us, get in touch with Ralph. If you have any questions, we will be sending out details of our next speaker and talk and date and things in our seminar series. I think we're planning to do 3 or 4 webinars across the year, kind of trying to address some of these sort of topics, giving out a bit of information about some of the science that we and other people do that's relevant to you and your families. So thank you very much. Thanks again, Ralph, and thanks everyone for attending.
Yeah, thanks everyone. Cheers. Bye.